Down in Petersburg, everything's fine. All them cats is drinking that wine. Drinking that mess is their delight. When you get drunk, start singing all night.
Hi, my name is Jordan Beaver, and I'm a PhD student at the University of California, Davis. I'm originally from South Carolina. I was born there and raised there, went to undergrad there. So when I graduated, I decided that it was time to try something new and explore a different part of the world. I majored in chemistry in undergrad, and I loved the research aspect of the field, but I really wanted to be able to work on more applied projects. UC Davis's agricultural and environmental chemistry group seemed like a fantastic fit for me because they give you the freedom to rotate into laboratories across many different disciplines. My first rotation just happened to be in the viticulture and enology department with Dr. Anita Overholster, and I immediately fell in love with the work I was getting to do. The Overholster lab does a fair amount of research on red blotch, a recently discovered virus that affects red grapevines. Only a few groups around the world are really looking into this virus, so it's very exploratory and new, which is very exciting to be a part of. Additionally, we've been collaborating with another lab on campus to study cell wall interactions with red grape pigment molecules, and this is what I'll primarily be focusing on over the next year. Our end goal is to collect massive amounts of data and use it to build a kinetic model for winemakers to use as a guide for controlling the hue and saturation of their wine. So I'd just like to say thank you to the American Wine Society Educational Foundation as well as the Banfi Vintners for supporting students like myself in pursuing their dreams. Because of their generosity and support, I'm able to continue working towards my goal of obtaining a PhD, all while doing research that I actually enjoy doing. So once again, thank you, and I hope you continue to support the research fields of viticulture and enology.
Get drunk, start singing all night, drinking wine, sport you to drink wine. Wine, sport you to drink wine. Wine, sport you to drink wine.
Hi folks, my name is Bryson Hill. I am the recipient of the East Tennessee Smoky Mountain AWS Chapter Scholarship. Right now we're in some of my research plots of Cabernet Sauvignon at Virginia Tech's Research and Extension Center in Winchester, Virginia. And here we're looking at two different canopies. That's what a lot of my research has to deal with, is vine size modification. And we're looking at a few different treatments for that. Those treatments being root restriction, under trellis cover cropping, and rootstock selection. So here on my right we're looking at an unrestricted root system. And as you can see, this is a lot more full, dense canopy. And over here, we're seeing a restricted root system using these fabric root bags. So there's some advantages to having a canopy like this. One of the main points to having a canopy like this is that you get more sunlight penetration to the fruit. And that results in better pigment accumulation. So we're going to see more color in those resultant wines, and also a higher degree of soluble solids. And we also have the added benefit of less canopy management, which a lot of grape growers can attest to. Labor is their biggest cost, so that's a big factor. And also we see less disease incidence in canopies like this. So that's a little bit of what I'm working on. I just want to thank AWS for all their support and awarding me with this scholarship. It means a lot to me, and I think it's really important for students like myself who are trying to pursue careers in this industry.
Pass that bottle to me. Drinking that mess is their delight. When it gets drunk, start fighting.
Hi, I'm Jay Martinock, and I'm a master's student in the Wine Research Center at the University of British Columbia in Vancouver, Canada. My research is focused on profiling yeast and fungal communities in British Columbian vineyards. Yeasts are extremely important to wine since they're the microorganisms responsible for performing fermentation, converting the sugars in grape must into ethanol and other byproducts. They also impact wine's sensory complexity and regional character, or its terroir. My research has two components. The first is a multi-year project looking at how the winery environment influences the Saccharomyces and non-Saccharomyces yeast populations from the vineyard. The second component is a survey of yeasts in vineyards across British Columbia's Okanagan Valley wine region. We're evaluating differences in yeast population composition and also searching for unique yeast species and Saccharomyces cerevisiae strains with desirable winemaking traits. We're in the first stages of this research, but we found evidence of indigenous yeast strains with fermentation potential, and we hope to find more as we expand our survey area. I've worked in the BC wine industry for over 10 years, both in the lab and as a winemaker. Our industry is young, and we have great potential as a world-class wine region. I'm excited to be involved in research that will help improve our understanding of BC's microbial terroir, and I hope too that we may find some unique BC yeast with novel traits that we can use to enhance our wine's regional character. I would like to thank the American Wine Society Educational Foundation, and in particular the Columbus, Ohio AWS chapter for the scholarship in memory of Nancy Gorey. I'm honored to receive this scholarship, and I very much appreciate your support of me and of wine research in North America. Thank you very much.
Delight. When it gets drunk, start fighting all night. Knock down windows and tear down doors. Drinking half gallons and calling for more. Drinking wine,
I'm Caroline Merrill, and I'm a PhD candidate at Washington State University. My passion for the wine industry is driven by the fact that it's a tight-knit community where almost everybody works together to drive the industry forward as a whole. This passion is only deepened by my love of working in a field where the industry benefits directly from the research being conducted, an area in which I'm on the ground floor. My research focuses on two of the most important quality indicators in wine: color and astringency. As grapes ripen, they accumulate more anthocyanins, which are the compounds responsible for color. Tannins, on the other hand, which are responsible for wine astringency, cannot easily be predicted by berry ripeness, but can be manipulated by winery treatments such as increased alcohol. Once extracted into wine, anthocyanins and tannins react to form polymeric pigments. These compounds are responsible for both the long-term stable color of wines as well as a decrease in astringency over time. My project looks at how both berry maturity and wine alcohol impact the formation and stability of polymeric pigments over time. In order to accomplish this, we harvest Cabernet Sauvignon and Syrah grapes when they are unripe, ripe, and overripe. Once the fruit is in the winery, we have additional winemaking treatments that manipulate alcohol. This research will hopefully enable winemakers to make picking and winemaking decisions with insight on long-term wine quality.
I would like to thank the American Wine Society Educational Foundation and the Bill Tuller, Columbia, South Carolina chapter of the American Wine Society for this scholarship.
drinking half gallons and calling for more. Drinking wine, sporty, you to drink wine. Wine, sporty, you to drink wine. Wine, sporty, you to drink wine.
Hi, my name is Megan Shoemaker, and I'm a master's student at Washington State University.
I came from Madison, Wisconsin, where I received my bachelor's degree in biology with an emphasis on microbiology. And coming from a state more well known for its cheese industry rather than its wine producing, I was excited to be working on something new. I am currently researching the wine spoilage yeast known as Brettanomyces, or Brett for short. Through Brett's metabolism, various precursors can produce several aroma and flavor compounds. These compounds are associated with aroma descriptors including smoky, sweaty, and barnyard, and in turn have the potential to severely alter wine quality.
In being a part of the sensory lab here at WSU, my research project will include evaluating the influence of the presence of 3 volatile compounds associated with Brettanomyces growth on the sensory properties of a Portuguese red wine blend.
This study, conducted in both Washington State and Portugal, evaluated aroma perception by a sensory consumer panel. By conducting the study in the 2 locations, geographical differences will be analyzed. A difference from control design was used to determine the magnitude of difference along a 9-point scale between a control wine and a test sample containing different concentrations of the 3 volatile compounds of interest, which are 4-ethylphenol, 4-ethylglycol, and 4-ethylcatechol. In addition, a questionnaire assessing wine and winemaking knowledge will be used to evaluate wine experience. With these results, consumers will be characterized into one of 3 groups, ranging from novice to expert.
Variances between these two populations may supplement past and future research by incorporating how geographical differences in wine expertise may influence the perception of Brettanomyces aromas. I would like to sincerely thank the American Wine Society Educational Foundation for awarding me this scholarship, and I'm extremely appreciative of the continued support provided by AWSEF.
Wine's for you to drink wine. Pass that bottle to me. Hoy! Drink it, man.
Hi, my name is Maria Smith. I am this year's recipient of the Southeastern Pennsylvania Regional American Wine Society Scholarship in honor of Larry and Eleanor Edmondson. Currently, I am a third-year PhD student in horticulture at Penn State University, studying viticulture with Dr. Michaela Centenari. The goal of our research is to understand how management practices affect cold stress survival in Pennsylvania-grown grapevines, as cold stress is often cited by growers as one of the most limiting factors to high-quality wine grape production. Towards this goal, we've developed 3 projects that examine 2 types of cold stress. The first is midwinter dormant cold stress, and the second is cold stress experienced during the growing season at early stages of phenological development. Excessive crop load can contribute to poor winter survival in cold climate regions. 2 of our projects focus on the use of early leaf removal at trace bloom and fruit set as an alternative method to cluster thinning for yield reduction in high cropping varieties, hybrid Chancellor and Vitis vinifera Gruner Veltliner. We evaluate a range of physiological responses as well as yield, fruit and wine quality, and wine sensory preference. To assess how crop load regulation affects vine winter survival, we take monthly samples to measure bud cold hardiness and tissue samples to monitor levels of carbohydrates associated with cold hardiness. Since grower cost is an important consideration for economic sustainability, we also collect data to calculate economic impacts on bottle costs in order to assist decision making. Our third project takes place here at Rock Spring, the research horticulture farm for Penn State located just outside of State College. With this project, we aim to assess how late spring cold stress affects new shoot growth in terms of mortality, loss of xylem function, whole plant functional recovery, and subsequent midwinter bud cold hardiness. This project was conducted on 2 vine varieties, Vitis vinifera Chardonnay and a new Minnesota cold hardy hybrid release Le Crescent. Both of these vines are 1 year old. That was a quick summary of my PhD research. We hope this information is useful for growers for making decisions on vineyard management practices, as well as selecting varieties for new vine plantings. Thank you for letting me take this opportunity to share my work with you.
Drink it, man. Oh, give me some of that slop. Oh, pass that bottle.
It is an exciting time to be learning about growing grapes and to be a part of agriculture in the state of Alabama. We're working hard on research that is shaping the future of food in the southeastern United States. Especially in the field of horticulture, we have several unique projects underway. Critical work is being conducted with recently patented Auburn kiwi fruit cultivars. Although our bubbling new hydroponic program is still in the seedling stage, it is bursting with promise. Deep in the heart of Alabama's peach country, there is a project stirring with the opportunity to change the face of southern vintages for years to come.
Hi, my name is Andrew Swantek. We're here in Clanton, Alabama at the Chilton County Research Extension Center. We're standing in our 87.5% Vitis vinifera, Pierce's disease-resistant selection vineyard. These 3 selections that I'm working with are coming from the UC Davis breeding program. They're able to sustain here in the southeast. And what I'm doing is I'm characterizing them for their vegetative traits, their fruit quality, and their phenological development. This is U0502-10. It's our first ripening selection and it has the largest clusters.
From Alabama's coastal plains through the Piedmont, limestone valleys, and up to the Appalachian Plateau, this state is thirsty for viticultural adventures, and these Pierce's disease-resistant vines are a gateway to new exploration. Learning to grow bunch grapes across the South has been an educational gift. I've been introduced to beauty and the beasts. In Alabama, along with the numerous fungal pathogens, entomological foes, and avian pests common to the eastern United States, we are also afflicted with Pierce's disease, a bacterial pathogen that causes drought-like symptoms in susceptible European wine grapes, visibly strangling the life out of them. It is readily spread by piercing, sucking insects such as the glassy-winged sharpshooter, which are prevalent throughout Alabama's riparian areas. The environmental conditions that result in the prosperous life of grape pests also present an enormous opportunity for future viticulture to thrive within new, unique geographical regions hidden throughout the South. As our researchers develop techniques for superior fruit production, the prospects of sustainably producing Pierce's disease-resistant vinifera bunch grapes appear bright. We will continue to improve growing healthy vines, making quality wines, and ensuring the future of agriculture is as sunny as the sky in June.
Down in Petersburg, everything's fine. All them cats is drinking that wine. Drinking that mess is their delight. When you get drunk, start singing all night. Drinking wine, sporty, you to drink wine.